Hello everyone, meet us again in another video. I'm Syafira and my friend.
My name is Fadilla Sherlyna.
I am Muhammad Ilham Fahri.
So last time we already talked about the wine productions, but today we will inform you about grape shoot culture. Now let me get into the introduction. Grapevine is a commercial fruit crop native to Eastern Asia, Europe, Middle East, and North America. The grape can be consumed as a fresh and dried fruit or used for making drugs, jam, and wine. But there's also a problem within their growth. Many serious diseases, including fungal, viral, and bacterial diseases, that are accountable for the low yield and shortened lifespan of grapevine. So, what is the solution? The answer of the problem is shoot culture, but there are also another concerns about the medium compositions, growth regulators, and concentrations, which is affecting the growth and in vitro propagation time of explants. Small changes in the concentrations can activate gene shifts, which may inhibit or initiate the metabolic process of the cell. So now we will investigate the way to optimize the level of various plant growth regulators in vitro cultures based on this article.
Next is the method for this research. There are many methods for this research, and this is the overall. There are three steps for this cell tissue culture. First, the explant culture. Second, culture establishment. The third, shoot multiplication. And there are two frequency calculation. First, callus induction, and the second, in vitro shoot regeneration. Next. So the step one, the explant culture, do the sterilization procedure before the explant grow in the media. So this is the key for this process. Rise, soak, for surface disinfectant and wash and collect part of apical meristem for this culture. This is the detail in the left show you about the flow chart for this method. The second, culture establishment. Do the cultivation in incubation procedure for the growth explant. So the key is we use the medium MS and not use the supply regulator growth. This is the flow chart for this stage. Next. The third is shoot multiplication. So after we get the explant before in the medium, we do the multiple cultivation for specific grow. It's mean the differential cell. So the key is grow multiple shoot and media MS plus supply regulator growth. So we want to get the specific, we have to supply regulator growth. This is the explanation about flow chart for this stage. Next. First calculation, the callus induction. The state of calculation callus induction for knower about influence of different concentrated regulator grow into media MS culture use this equation. After we get the callus, we have to calculate the size of the callus. We use this equipment. Sorry. And last, we have to calculate about the in vitro shoot regeneration. The measure the shoot regeneration for the subculture that already growing in media. Use this equation. This is the flowchart for the detail for this step. Next.
So the next one is the result, which the balance and appropriate concentration is required to get the desired product. Next. The first one is to establish in vitro environment. To initiate in vitro culture, surface disinfection needs to be done to avoid fungal contamination of grapevine explants. In this study, shoot tape explants were disinfected with 0.1% mercury chloride for 7 minutes. Then under aseptic conditions, meristems were excised and cultured on nutrient medium. The result is, if we compare to other plant tissues, apical meristems contain less endophytic contamination and grow well when refined in vitro. Next. The next one is the growth of in vitro shoot cultures. The shoot number was actually affected by the different combinations of growth regulators. In the shoot proliferation, a nodal segment containing an axillary bud was taken from the in vitro grown shoots and cultured on half-strength MS medium along with the growth regulators combinations. The combination with the highest number of shoots per cultured explant was benzylaminopurin or BAP and gibberellic acid or GA3. It could be concluded that the addition of BAP has a huge impact in the development of axillary buds and shoots in explants, while the combination of GA3 with glycine produced less than the one cultured with BAP. Also, the product was distorted. Cytokinin can indeed improve shoot multiplication, as BAP can produce much more products than other growth regulators. However, in the shoot length, as the BAP concentration increases, the length becomes shorter. It might be due to its toxicity. As for the combinations of kinetin and glycine with GA3, it resulted small and distorted shoots. Hence, the combinations of growth regulators and their concentrations have to be in control as they produce different results. Next. For the shoot regeneration, a significant different number of shoot was formed in response to combinations of BAP and NAA. There are four different combinations. First is without growth regulators, which got the lowest shoot regeneration frequency of 6.67%. The second combination was 0.5 milligram per liter BAP plus 0.2 milligram per liter NAA. That an increase in the regeneration frequency of 16.67%. For the third combination, 1.0 milligram per liter BAP plus 0.3 milligram per liter NAA was also increased with 26.67% for the regeneration frequency. Last and the best one for 1.5 milligram per liter BAP plus 0.5 milligram per liter NAA combination, the maximum shoot regeneration frequency of 53.33%. Hence, it can be concluded that the higher concentrations of BAP and NAA increase the regeneration rate. Also, the shoot regeneration of grapevine from calluses of leaf origin is sensitive to auxins and cytokinins. A specific concentration is needed for optimum shoot regeneration. The regenerated shoots have firm roots when moved to half-strength MS medium without any growth regulator and kept at about 25 degrees Celsius. Next. As for the callus induction, in order to proceed with the callus induction, leaf discs from in vitro shoot cultures were used. They were placed on MS medium supplemented with plant growth regulators. In this case, R2,4-D, BAP, and alpha-naphthalene acetic acid, or NAA, which differ in the concentration. While for the NAA is used at constant rate of 0.2 milligram per liter, high level of 2.4-D, which is 2 milligram per liter, with the lower level of BAP, which is 0.3 milligram per liter, yielded the highest number of calluses, which is 73%. In contrast, the lower level of 2,4-D, which is 1 milligram per liter, combined with the higher level of BAP, resulted in lower callus induction frequency, which is 31%. As for the control treatment, it induced the lowest frequency of callus induction, which is 6.66%. The resulting condition might be caused by the lack of optimum auxin concentration needed for the induction. This resulting study shows that a high level of 2,4-D can induce more calluses from leaf discs. The higher level of 2,4-D combined with lower level of BAP and constant NAA produce callus formation and make the formation rate faster. Consequently, NAA and BAP didn't stimulate the excellence growth and callus formation when constructed with 2,4-D. Still, callus induction is positively affected with the modest quantity of BAP, which is 0.2 milligram per liter. Next.
So after all this process, we can conclude that for the routing of regenerated shoots, recommended to use half-strength MS medium with no growth regulator. And this is our references for today's presentations. What do you think about culture shoot? Shoot culture, sorry. Shoot cultures, comment us, and don't forget to like our video. Thank you, stay healthy and happy.